Hi, and welcome to the world of macromolecules. There are four main macromolecule categories, as listed here. In this video, we will deep dive into the first two, carbohydrates and lipids. Now, here are some images below that are showing space-filling models of these four classes of the biologically important molecules. We can see the carbs, the lipids, protein, and nucleic acids in their three-dimensional configuration. We have different colors representing the different elements and their atoms. And I think that this is a great overview of their complexity. And we might not appreciate it right now, but I think this is a great slide to jump back to at the end when you're reviewing for this section, because it brings a lot of their structural differences to light. So let's first start with carbohydrates. Carbohydrates can be classified really basically as simple carbs, which are called monosaccharides, and sometimes disaccharides are still quite simple in their nature and structure, versus complex carbs, which are called polysaccharides. So carbohydrates have the basic chemical structure or formula of CX, H2X, and OX. So there's always going to be carbon, hydrogen, and oxygen that create this molecule. And the carbon, hydrogen, and oxygen will be found in a 1 to 2 to 1 ratio. So let's use the example of glucose. And glucose has a C6H12O6 chemical formula, which obviously coincides with the 1 to 2 to 1 ratio. Now, monosaccharides are the most basic unit of a carbohydrate. It's the smallest building block, and that's why it's called a simple sugar, these monosaccharides. When we talk about simple building blocks, the tiny pieces that come together to actually form the macromolecule, we call those monomers. They're the most basic building blocks that then can join together to create more complex molecules. So monosaccharides include sugars that you've probably heard of before, such as, well, hopefully you've heard of glucose, also fructose and galactose. These are simple sugars. So they could be classified by the number of carbon atoms that they contain. I have a little table there, and if they have 3 carbon atoms, it's a triose, 4 would be a tetrose, 5 a pentose, and 6 a hexose. So here are some examples. You can see on the first side there, on the left, we see a 3-carbon monosaccharide, or a triose. Now, the one in the middle, sometimes we'll be seeing, quite often actually, we'll be seeing diagrams of molecules, not just of carbohydrates, but of different macromolecules that don't necessarily indicate all of the atoms or elements present. It's insinuated that we understand it. So let's just take a look here. See how there are kinks along, which are representing bonds. Where there are angular kinks, that's actually showing where a carbon would be. So I put the carbon in there, the red C's, okay? But I want you to start to get to understand that we won't always be seeing carbons. There's just carbon everywhere in so many molecules that it can really crowd it all together. So oftentimes they'll focus on putting the main ideas and the functional groups, and they'll leave the carbons out, for example. So this one, the second one in the middle, is also a triose sugar. And then those two are shown in their linear form. And then the third one on the right there is showing its cyclic form or ring structure. And again, wherever there's a bend or an angle, that implies that there's a carbon. And in this case, there's 1, 2, 3, 4, 5. So pentose. Okay, so as I said, they can either be present as a long chain or a ring structure. Most commonly, monosaccharides are cyclic or ring structure when they are placed in aqueous solution. They tend to form that ring. The ring structures that form vary depending on the location of those hydroxyl groups at carbon 1. Okay, so I labeled carbon 1 down below on both the alpha glucose molecule and the beta glucose molecule. And the way that we typically number these carbons, because they won't often be numbered, but we'll start on the carbon on the furthest right of the ring, and then we'll go down and clockwise to the top and even up if there's any carbons up at the top there. Okay, so I follow that pattern for both ring structures of these monosaccharides, and you can see that there's a hydroxyl group bonded to carbon 1 in alpha glucose, and it's pointing down. It lies below the ring. So that is what characterizes this compound, not just as glucose. Both of these compounds have C6H12O6 chemical formula, but just the arrangement of the hydroxyl group pointing down, it slightly changes certain properties of it, and we call that alpha glucose. Now, if you look at the one to the right of it, it only differs by the hydroxyl group on carbon 1 pointing upwards. So it lies above the ring. We call this one beta glucose. Okay, so the hexose carbohydrates, those with 6, include glucose, galactose, and fructose. These molecules all have the same chemical formula. So C6, that's why it's hexose, 6 carbon, H12O6, but their physical arrangement differs. So oftentimes we refer to glucose as C6H12O6, but actually you can't confirm that until you look specifically at its structural composition. These 3 molecules are all isomers. They're still monosaccharides. They're just a ring in themselves. They're not bonded to each other here. Okay, they're just showing the 3 separates. And I threw down the definition of isomer there. These are molecules that have the same molecular formula, such as beef, but based on where different, perhaps functional groups, in this case, we see hydroxyl groups differing quite a bit between the 3. They have a different arrangement of the atoms in that space. Now, on to the disaccharides. So previously that was monosaccharide. You only saw one ring structure, okay? One monomer, that simple building block, that one simple sugar. Now, disaccharides, too, that prefix di meaning 2, are 2 monosaccharides, those basic building blocks. 2 of them are linked together. And we know that molecules can be linked together through a condensation reaction where it loses 2 H's and an O between the 2 molecules bonding together, and water is lost through that reaction. And what we get is disaccharide in this case. We get a larger molecule. Okay, so sucrose is a good example of a disaccharide. This is formed when there is a fructose molecule, and through condensation reaction, it bonds with a glucose molecule. Now, maltose is another disaccharide, which is shown below, and that is the product of this reaction. And it is formed when a glucose molecule and a glucose molecule, so 2 glucoses, join together. Again, this is through condensation reaction. You could see through this arrow that's implying that the reaction is taking place, that water is lost. Okay, obviously we know that a larger molecule can be broken apart too through the hydrolysis reaction. So this is showing the opposite. This is not how disaccharides are made. This is how the body breaks apart, or through catabolic metabolism, breaks it apart into its individual monomers again. Okay, and with this reaction, water is required with that larger disaccharide to be able to lysis or break that glycosidic bond that is holding the two monosaccharides together. So I put their glycosidic bond. That's actually when condensation reaction happens and those two monomers or monosaccharides bond together to form that disaccharide. That's what's holding it together. We call that, for sugars or for carbohydrates, a glycosidic bond. All right, now we're on to the polysaccharides. Now, poly means many. And in fact, so many, it's hundreds upon hundreds of monosaccharides linked together. While monosaccharides represent simple sugars, these polysaccharides are our complex sugars. Polysaccharides are polymers made up of hundreds of monomer units of monosaccharides. Okay, so remember monosaccharides, those simple rings, not only are they bonded to another simple ring to form a disaccharide, they're bonding hundreds and hundreds of times to make long chains. Polysaccharides can exist as long chains, or they can even have the long chains with branches coming off of them. We'll look at a few examples of that. The shape and linkage determines the physical properties of each polysaccharide. So are they just simple chains? Are they chains with branches? Can these chains line up parallelly and have intermolecular forces holding them together? We'll take a look. Some biologically important polysaccharides include cellulose, which you could see there on the right, starch. These are found only in plants. And then animals produce glycogen, which is found in animals. Starch and glycogen are storage units, and cellulose provide structural support for plants. We find those in the plant cell walls. All right, let's go to lipids now. So oftentimes we think about lipids as fats, which is correct. However, there's more categories than just fats itself that lipids comprise of. Lipids are macromolecules that provide long-term energy storage, similar to glycogen, insulation, cushioning of internal organs. Our organs do have slight fat around them so that if we get jostled around, we get punched or we fall, there is a protective layer around them. Again, too much is not good, but a little bit is great. They are the main components of the cell membrane and are the main structure of hormones. So all lipids are hydrophobic, meaning that they do not dissolve in water. And we know phobia, fear, do not like, and hydro meaning water, so hydrophobic. And we could see there on the right, that's a picture of olive oil probably being thrown into some water, and we call this being immiscible, where that fat, for example, is not able to break apart and dissolve properly to create a homogeneous solution, which is why it's immiscible. So there are four main types of lipids. So oftentimes we'll think of fats, but there's also phospholipids, steroids, and waxes. So let's first start with fats. So fats are the most common energy storing molecule in an organism. In fact, 1 gram of fat contains 2 times as much energy as 1 gram of carbohydrate. Most of the excess glucose that is not converted into glycogen is then transformed into fat. The most common fat is the triglycerols, or we sometimes call them triglycerides, which contain 3 fatty acid chains attached to the glycerol backbone. So on the image on the right there, we can see these parts as separate at first. So the glycerol backbone there's 3 carbons and there's 3 hydroxyl groups, and then the rest are hydrogen atoms. And then we have the fatty acid Sometimes they're called fatty acid tails or fatty acid chains. And you can see that it's pretty much a long chain of hydrocarbons, more or less. Now, they bond together. And when this bond occurs through condensation reaction, an ester bond is formed. So it's not a glycosidic bond, which forms during condensation reactions to combine carbohydrate molecules. When we're talking about lipids, this is an ester bond. And again, you can see on the right there that water is released for each bond that is formed because this is the nature of the condensation reaction. So here's another image. This slide pretty much says what the previous one did, but it's showing a bit more structurally sound molecules. You can see all the players in action there. Okay, so you can see that the glycerol has the hydroxyl group and the 3 fatty acid chains. Each one has a carboxyl group, and these are what link together to form that ester linkage there in the triglyceride. Now, it's showing that this could go both ways, just like the carbohydrates. So we can use the monomers. In this case, the monomers, the basic building blocks for triglyceride is the glycerol and the 3 fatty acid chains, right? Those are the basic building blocks. And through condensation, they could bond together. And that's the arrow in the blue at the bottom showing this reaction moving towards the right to form that more complex molecule. And then hydrolysis can also happen to that more complex molecule, that triglyceride, where water is put into the system. The molecule takes the water and it helps lyse that larger molecule back into its individual monomers, the glycerol and the 3 fatty acid chains. So let's talk more about fatty acid chains. There are 3 types. There are saturated, unsaturated, and polyunsaturated. So saturated means like maximum capacity, right? So these saturated fatty acid chains contain the maximum number of hydrogen atoms per carbon atom. So remember, each little bend in that fatty acid chain, if you're looking at the pictures below, each little bend has a carbon atom. Okay, so each carbon atom only has 1 bond to each other. You can only see 1 line there for the saturated fatty acid chain there. That means that the other 2 bonds that I can form with are with hydrogens on either side of that chain. Okay, now red meat, butter, and sweets have this type of fatty acids in them. And because they don't have any double bonds, because they are just singularly bonded to each other, this makes them quite flat in nature. Although you can see kinks, it's actually quite flat and they're linear. So they could stack up on top of each other fairly easily, not leaving too much space. And that allows them to be solid at room temperature. Now, the problem with that is that these fats can increase cholesterol, which is associated with plaque buildup in our blood vessels, which then can lead to heart disease. So I put a sad face there. These are the bad fats. Then we have unsaturated and polyunsaturated, so multiply unsaturated carbon bonds. So when we're talking about saturated and unsaturated, again, it's all about the carbon bonds, okay? How many hydrogen atoms can these carbons bond to? And if we look at this second chain here, the unsaturated, you could see that there's a little line halfway down that chain that is right on top of the main lines. So this is implying a double bond between those 2 carbons. Now, the problem with a double bond is that takes away the bonding capacity for extra hydrogens on both of those carbons involved in the carbon bonds. So this creates an actual, a little more kink into a slightly more bent chain, not by too much, but just a little more. And what that does, they can't stack perfectly on top of each other. And this leads to them being liquid at room temperature. These guys are actually good because they are able to decrease cholesterol in the body. So these are the good guys. They are found in olive oil, avocado, and nuts. And then we have polyunsaturated. So not only are there double bonds once along that chain of fatty acids, we could see that there's a double bond. They've actually numbered these at the carbon 7, carbon 10, 13, 16. You can see all along the bottom there, there's double bonds. So this makes them very kinked or bent in many different spots. Again, they can't line up well. They're liquid at room temperature, and they also decrease cholesterol. So these are good guys as well, and they are found in fish, like the omega-3s that we talk about, poultry, and canola oil. Another category are phospholipids. So phospholipids, these guys are the main component of the cell membrane. We can see that there are two parts involved. It's a quite simple type of fat. There is a hydrophilic head and a hydrophobic chains. So the hydrophilic heads, these will be sticking outwards of the cell membrane, either into the extracellular matrix or to the intercellular matrix. And the hydrophobic tail will be really repelled away from any aqueous solution. So they will be kind of sticking into the cell membrane itself, where it is not an aqueous solution. Another category of lipids is steroids. So steroids are an important group of lipids that consist of 4 linked carbon rings. Steroids are hydrophobic and include cholesterol and cortisol, as well as hormones such as estrogen and testosterone. So steroids are naturally produced in the body and are necessary for proper growth and development. Hormone supplements that mimic the naturally produced hormones in the body are commonly used by athletes who would like to gain more muscle strength, leaner body mass, or speed. So the type of steroid that they would be using are anabolic steroids, which we know anabolic means building up. So these steroids would specifically be building up the muscle tissues. You can see down below, here's a couple examples: testosterone, cortisol, progesterone. These are steroids, and all of them have 4 linked carbon rings. So that is, if you see 4 linked carbon rings bound together like this, in a hexagonal arrangement, each of those rings are, then that would be a steroid. You're probably aware that the use of steroids is banned by pretty much every professional and amateur league, and including the Olympics. So yes, it's trying to make our bodies do something that naturally it is not, and it's just not fair if other people are not using steroids to help them and others are. Of course, now they're getting very sneaky with the ways that they try to enhance their performance in sports. So I'm sure you've heard of blood doping, which is EPO, and what they have been doing, especially in cycling. I think this is pretty big because they have, the Tour de France. They're cycling up mountains for 40 days. They before the race, like a month before the race, they remove quite a bit of volume from their red blood cells, and their body has to reproduce red blood cells to make up for the loss. They store those red blood cells, and then on the day of the race, they inject it back into their body. So they now have extra red blood cells, and we know that red blood cells are able to transport oxygen efficiently through our body, which allows for better muscle performance, right? So that's what's happening, which I guess they're using their own body to do so, but it's in a very manipulative way. So for a long time, they were not able to detect this. And it wasn't until the Lance Armstrong fiasco in the Tour de France where they thought, wait a second, we need to find a way to better test for this. So now they can actually test for this. But again, there are doctors out there who are working with endurance athletes, trying to find other ways to allow them to peak in their performance. All right, cholesterol. So cholesterol is also needed by the cells of the body. Cholesterol is produced within the body and can also be attained through diet, although too much cholesterol causes problems for the circulatory system. Cholesterol is needed for the cell membrane to function properly. It can also be converted into compounds such as vitamin D with the use of sunlight and bile salts. So often when we talk about cholesterol, cholesterol typically gets a really bad rap. We think of the bad cholesterol, which is actually called LDL, low density lipoprotein. And this bad cholesterol carries cholesterol to your cells. It carried it directly to your cells. But when you have too much LDL, it can build up in those artery walls, right? In those blood vessels that carry blood to your cells, causing them to narrow. This reduces blood flow, which can be bad for your heart health. Now, luckily, we have the superhero cholesterol, the good cholesterol, the high density lipoprotein, the HDL. And this good cholesterol carries excess cholesterol in your blood back to your liver. So it's not carrying it to your cells where it can build up in plaque. It's taking the extra cholesterol and it's bringing it to the liver where it's broken down and actually removed from your body. This means a high level of good cholesterol can maintain your heart health. And let's not forget about waxes. So waxes are composed of long chains of fatty acids that are attached to an alcohol or a carbon ring. So you can see down below, there are some rings. They're not attached right beside each other like steroid. And it doesn't have to be 4. It could be different numbers. Here there's 2. And there's also long chains of fatty acids between them. A plant's epidermis cell produces cutin, or you may have called it a cuticle on the upper epidermis. This is a thin, clear coating that covers the leaves as well as stems and fruit. So the cuticle is really important because it prevents water loss or evaporation from the plant. It also protects the plant from infection. Bees also produce a wax that is used to form their hives. Beeswax is used commercially in both cosmetics and pharmaceuticals. There's also carnauba palm trees that are used commercially to use that wax for polish for shoes, floors, and furniture, along with cosmetics and the paper industry.